Hello, good afternoon. So today we will be jumping on another topic, and that is about enzymes. For this course or this topic, we will be doing definition, or we will be discussing the definition of enzymes, enzyme function, classification of enzymes, mechanism of enzyme action, and lastly, factors that affect rate of enzyme activity. So the learning outcomes should be number one, describe the structure and functions of enzyme. Second, explain the basic principles of naming enzymes. Third, differentiate chemical properties of several types of enzymes. And then fourth, explain the factors that affect enzymatic activity. So every chemical reaction needs a certain amount of energy to start. We call this the activation energy. Think of it like pushing a ball up a hill. Once it's past the top, it rolls down easily. So catalysts like enzymes lower this activation energy, making reactions happen faster and easier. This is why enzymes are essential for life. Without them, most reactions in our body would be very slow to sustain life. Um, Enzymes are biological catalysts made of proteins. They speed up chemical reactions in living organisms without being used up or changed permanently. So, what makes them special is their specificity. Specific. Each enzyme works only on one kind of substrate, like a key fitting into a lock. So we have types or classifications of enzyme based on the type of reaction they catalyze. So for example, um, oxidoreductases for reduction reactions, transferase for transferring functional groups, hydrolases for breaking bonds using water, lyases, isoreses, and ligase, each with their own roles. And this classification helps us understand which reactions they could control inside the cell. So enzymes work through the enzyme substrate complex. So the substrate binds to the enzyme's active site, like a puzzle piece fitting perfectly. The enzyme then helps the reaction take place by lowering activation energy. After the product is formed, the enzyme is released unchanged and ready to be reused. So models like the lock and key and induce fit explain this process. Okay, let's go now to factors affecting enzyme activity. And first is the temperature. Enzymes are sensitive to temperature. As temperature increases, enzyme activity also increases up to a point. But too much heat can denature or destroy the enzyme structure, stopping its function. That's why our body maintains a stable temperature so enzymes work at their best. Next will be the pH. Each enzyme works best at a specific pH level, called the optimum pH. So, for example, pepsin in the stomach works best in acidic condition. Pepsin is the enzyme to process protein, while amylase in saliva prefers a neutral pH. Amylase is the enzyme in charge of digesting carbohydrates. If the pH is too low or too high, the enzyme's shape changes and it won't function properly. Another factors that affect enzyme activity is the enzyme concentration. The more enzyme molecules present, the faster the reaction rate because more active sites are available for substrates. But if the substrate is limited, adding more enzymes won't increase the rate any further. It is like having too many workers but not enough task to do. And lastly is the um, substrate concentration. So when substrate concentration increases, enzyme activity also increases because more substrate molecules are colliding each with other enzymes. However, once all enzyme active sites are occupied, the rate levels off. This is called enzyme saturation. Okay, so that's all for today for enzyme, and then I'll see you again. Goodbye.